Hello, my name is Jennifer Balacco. I'm from the Vertebrate Genome Lab, and today I'm going to be sharing with you some of our adventures towards generating error-free genomes. The Vertebrate Genome Lab is located at the Rockefeller University in New York City. We specialize in long-read sequencing technologies. Our expertise spans from sample collection and preparation to sequencing data production, genome assembly, and comparative genomics. We generate multiple different data types for generating high-quality genomes, including long-read sequencing with PacBio and optical mapping with Bionano Sapphire. We're involved in several projects that require these technologies and high-quality reference genomes, such as the Human Pangenome Project, and we serve as one of the main data production and assembly hubs for the Vertebrate Genomes Project. The Vertebrate Genomes Project, or VGP, aims to generate near error-free reference genome assemblies of all extant vertebrate species. This is a large project that includes over 150 members globally with the goal of gathering and generating these genomes in an open access genome repository. We're doing this in 4 phases. We're currently in phase 1, which looks to generate genomes for 260 species, representing all vertebrate orders with a divergence time of about 50 million years ago. In doing so, we're combining multiple different data types from long reads with PacBio and optical maps with Bionano to shorter reads of linked reads and ARIMA Hi-C. We look to also define the assembly's quality by looking at metrics such as contiguity, completeness, assembly accuracy, and base accuracy. The VGP sets some minimum metrics for these different measures, such as an N50 contig of about 1 megabase at least, an N50 scaffold of at least 10 megabases, a 90% of the genome assigned to chromosomes, a QB40, and some degree of phasing. So to get to a high-quality genome is this winding path that involves multiple steps before you get to the end product, from sample collection and storage to DNA extraction, data generation, assembly, and annotation and curation. Finally, you end up with a high-quality genome, which in and of itself is highly valuable, but also for downstream analysis. We see that sample quality greatly influences DNA quality, which then influences the data quality that can be produced, and then finally the assembly and final genome quality. Therefore, we spend a lot of time in looking at how to best extract ultra-high molecular weight DNA. Now, we think of ultra-high molecular weight DNA as something that has a high proportion of very large fragments. So as you can see in this pulse field gel, we're seeing the fragments above 300 kilobases all the way up into the megabase range. And you can see this short clip of me pipetting this highly viscous concentrated DNA sample with a wide orifice pipette tip, because this type of DNA sample sometimes can be very viscous. So to achieve that high quality of DNA, we can recommend certain sample types depending on what taxa to use for DNA extraction. For instance, across the board, we see that cultured cells work really well for generating high-quality, ultra-high molecular weight DNA at high yields. However, for certain taxa, we can recommend other tissue types, such as for mammals, we see great yields and fragment length from spleen. And from birds, we see the same for blood samples, and we really recommend blood stored in 95% ethanol. We also see that certain tissues worked best for transcriptomics when extracting for RNA, but these are all tissue recommendations that you can be found on our website. Also to be found on our website are the sample collection protocols. So it's not enough to just think this is the type of tissue I want per taxa, but it's also important to think about how to preserve that tissue to preserve the integrity for DNA extraction. One example of this is we recommend flash freezing with liquid nitrogen, but we know that for field applications, liquid nitrogen is not always easy to come by. Therefore, we have this very cool protocol that looks at using an ethanol dry ice slurry to flash freeze instead of liquid nitrogen, and it does so very effectively. Which brings us to the actual DNA isolation. When you need a high yield of ultra high molecular weight or high molecular weight DNA, certain things need to be considered for this DNA isolation, such as keeping a cold temperature when dealing with your tissues before lysis. Also, careful mixing with wide bore pipettes and being careful when vortexing is used. Also to consider are modifications per tissue type. So not every tissue type should be treated the same way for DNA extractions. I'll give you a few examples of this. For instance, if we're doing a DNA isolation with mammalian blood, now mammalian blood, the red blood cells are not nucleated. So as our example here, we did a DNA extraction from whole blood from a California sea lion using the nanobind CBB kit. We had an input of 200 microliters whole blood, and our yield was 7 micrograms of high molecular weight DNA. When we then move to a nucleated blood sample, so something like a bird, or in this example, a shark, their red blood cells are nucleated, so you actually need to adjust the protocol to a much lower input. You need much less starting material to achieve high yield DNA. Here our example looks at the epaulet shark, which is a very interesting evolutionary shark to look at, where we adjusted our protocol down to 7 microliters whole blood input and had a yield of 16 micrograms ultra high molecular weight from only 7 microliters of blood using the same nanobind CBB kit. So these are things to consider because if you're working with nucleated blood, you really don't need nearly as much as mammalian blood. So this is important for your protocol as well as for your collection. Now, one thing to consider with nucleated blood is that when you adjust your protocol of input, to be cautious when dealing with higher than 10 microliters per prep because you can overload the disc in the prep. Here you can see that the nanobind disc can absolutely bind very large amounts of DNA, but it makes it hard to effectively wash. It is possible to wash large amounts that are bound, but it is better to avoid overbinding and ending up with a less pure DNA sample. So this is just one of our examples of how knowing what type of tissue you're working with from what type of organism is very important when moving on to the actual DNA isolation because adjusting the protocol will lead to a higher quality, high molecular weight DNA sample. Which brings us to an example of one of our VGP organisms that we didn't know nearly much about that we would normally like to know. Here we have the Maguire stork. This was a museum specimen from the Smithsonian National Museum of Natural History. This tissue was collected in February 1992. Most of the samples we work with are collected much more recently to the date of DNA extraction, but this was all the way back in 1992. The tissue type was unspecified, so we know it was some sort of solid tissue type, but we did not know exactly which type, which is normally the type of information that we use to adjust our protocol when going on to DNA extraction. So we went and we extracted ultra high molecular weight DNA with the nanomide tissue big DNA kit. And sometimes when we try a museum specimen, especially older ones or ones we know less about, most of the time we don't achieve a high enough yield or that DNA ends up not being large enough fragment length. It ends up looking degraded. In this case of the Maguire stork, this specimen produced ultra high molecular weight with enough yield to move on to sequencing. Which brings us to some of our sequencing data. So for this case, we used PacBio continuous long reads, and we had the longest subread of 259 kilobases and a subread N50 of 23 kilobases. This is from a total PacBio CLR yield of 86 gigs and a coverage of about 72x for the genome size. If we look at our data from the BioNano optical mapping, we have 400x coverage from running a single Sapphire chip with a longest read of 2,800 kilobases, which is extraordinary, and a read N50 of 265 kilobases. We also generated 10x linked reads and ARIMA high C data for this specimen, and you can really see across the board that high quality DNA leads to high quality data, and sometimes it's even possible when a sample is old or when you don't know everything about it. With this data and this extraordinary group of people working together, it went into the VGP Genome Assembly 1.5 pipeline, which starts with the PacBio assembly and then goes through 3 levels of scaffolding, 3 levels of polishing, and a final curation, which is now our VGP Genome Assembly pipeline. Slightly different, but for the Maguire Stork, this is the exact one used, and these were some of the people who worked on it. From here, we can look at the final curated assembly, which used the same tissue sample as the previous reference genome of the Maguire stork. And we can see that when working with a high quality DNA sample and with the technological advancements, we see a great improvement on the contiguity and error rate of the genome. So we see contig N50 of 23 megabases for a total number of 384 contigs, and a scaffolds N50 of 85.5 megabases, total number of scaffolds being 152. And this is for a total assembly size of 1.2 gigs, which matches the previous assembly size. This curated final assembly also had a 97.8 complete BOOSCO score with a 97.1 complete and single copy. Earlier I mentioned having a chromosome scale assembly for VGP, and here the curation team looked at a high C, as we can see from the high C map, as well as the karyotype, where they were able to match up all 11 macrochromosomes and the W and Z chromosomes, the W in 2 parts. These were all identified based on our scaffold lengths, which is really great to find all of those macrochromosomes and sex chromosomes, as well as 18 out of the 26 microchromosomes were also assembled, characterized. So that was one museum specimen where we now have a final curated assembled genome, but we're in the works of more museum specimens and constantly trying to learn more about them, as well as learning more about low input samples, invertebrate samples, and all other different types of challenging vertebrate and non-vertebrate samples, which leads to further optimization based on sample type and potentially automating certain processes along the way as we improve technologically for generating near error-free reference genomes. With that, I'd like to recognize that all of this is a global team effort that involves many people, with much gratitude to everyone part of this team as well as sponsors along the way. So thank you for listening, and I suppose it is time for questions. So thank you.
Hello, everybody. Thank you for joining us. I'm here with Jennifer Balacco, who you just heard speak. My name is Nick Cisneros. I'm the director of the North American FAS team, and we're here to answer any of your questions. So we already have a few that have come through here, and I'm just going to read these off, and then I'll turn them over to Jennifer so she can answer them. The first one is, have you tried any microbial genome DNA extraction using high molecular weight DNA extraction kits?
So that's really interesting. We haven't here at VGL worked with microbial, or at least not intentionally, but I could see looking into that.
Yeah, I think that the kits are probably more specifically made for each organism, just due to like input amounts would be my guess. So that's a good question though. One of the other questions that I have here is, what are the minimum DNA quality standards you would consider usable for sequencing?
Yeah, that is a great question and kind of really depends on what you want to do, what type of sequencing you want to do, the size of the genome of the organism you'd like to sequence. So if you're looking to do something like PacBio Hi-Fi, you're going to need at least 15 micrograms of DNA, and you're going to want it to be high molecular weight, but you don't necessarily need it to be ultra high molecular weight DNA. Meaning, for something like when we do bio-nano optical mapping, we'd like our DNA to be within the megabase range of fragment size. So it really depends on the technologies you'd like to pursue and exactly which organism.
Okay, perfect. Thank you. And then one of the next questions here is looking at the huge improvements between the previous reference genome and your final assemblies, do you anticipate that you'll have to go back and resequence many of these specimens?
That's very interesting. So yes and no, it really depends. So we're constantly making these big leaps forward in technologies. So what we've done, 10 years ago, we can do better now, and then 10 years in the future, we'll be able to do better than what we're doing today. So depending, sure, of course, we'll always be improving. Our assemblies are never really done. They're never really perfect. So we can always improve upon them.
Okay, and then I have a few more questions here. Have you tried hi-fi library prep of DNA extracted from saliva?
Interesting. No, we have not. I could imagine that that would work really well, though.
Yeah, I think that for some of the COVID stuff that they were extracting from saliva and getting that done. Um, so I think that that is a possibility. And then I have one here from Andrew Crawford that says, wow, I probably missed this, but how was the mysterious and 28-year-old Magurai stock sample preserved and stored? Do we know?
So we do know that it was kept in, so no preservation liquid. So it was a solid tissue with no preservation liquid. And we do know that it was kept in cold storage, but I don't know the exact temperature fluctuation. So we've had it in our -80 for a few years now, but I'm not entirely sure with before that.
Okay. Oh, perfect. And then it looks like we have one last question here. It seems the very high molecular weight DNA is difficult to handle. Whoops, sorry, that skipped. For many applications, PacBio Hi-Fi, certainly such megabase scale DNA is not required to start out with, question mark. So essentially, do you need 50 megabase size DNA to start out with?
Yeah, you don't necessarily need it for everything, but there are ways to take this kind of unruly ultra high molecular weight DNA and help make it a little bit easier to handle. So there are dilution techniques, there are shearing techniques that can be done, and there are ways to kind of bring it down a little bit in size while still, if you extract a really nice quality ultra high molecular weight sample, you have an opportunity to use a lot of different technologies on it. But if you start at a lower fragment length, then you really limit yourself and you can't do those technologies that require the higher fragment length. So if you, let's say, only want to do hi-fi sequencing, then no, you don't necessarily need the megabase length DNA.
Okay, perfect. And then I have one here that says, what steps in extraction and library prep affect N50 values the most?
Oh, that's a great question. So I think definitely as far as sample preservation and DNA extraction, the quality of the DNA that you have absolutely affects the reads that you get once you do that sequencing, which therefore affects your assembly. So starting with a very high quality sample that was preserved and then using an extraction method that gives you high quality DNA will definitely affect significantly downstream what your N50 is going to look like.
Okay. And then I have one very last question. We have about a minute left, everybody, and then our time's gonna be up. The last question is, the circulomics nanobind protocols have a thermomixer step that's at 900 RPM for 30 minutes. How does that not break down the ultra high molecular weight to smaller sizes?
Yeah. So for the CBD of CBB, so for the blood protocol, you actually have two options and you don't necessarily have to use that thermomixer step. So in that case, you actually have a lot of flexibility. Interestingly, at that 900 RPM with the thermomixer, you still do get pretty high molecular weight DNA, we still see up to like 300 kb. But there are the protocols for ultra high molecular weight where you actually don't use the thermomixer at all.
Okay, perfect. All right, Jennifer, well, thank you so much for your presentation and for getting on the live Q&A with us. That was fantastic. And everybody else, thank you as well. There is a link at the bottom of the slides, this last slide here, closing slide, that if you could go on and give us some feedback, that would be appreciated. Plus, there's giveaways associated with that. So thank you all for your time today, and have a good rest of the day, evening, or morning.
Thank you.